right. Welcome everyone to the last edition of the Physical Properties of the Cytoplasm seminar series. The last edition before the summer is not the last edition at all. We already have more speakers lined up for the fall on potentially in August 19th or in September 2nd. We'll have the next edition. So you can already look forward to this after today's exciting talks. And then for today, I realized that my name was on the calendar. I shortly panicked, but then I was super happy because actually we have a lot of really cool projects on this topic. In particular, we are involved in many collaborations that concern the cytoplasmic properties. And so I thought today, since it's the last time before the summer, maybe we can do something special and have two talks. And so today we will have first a talk by Marina. She's a PhD student in my lab. And she will talk about a collaborative project that we are working on in collaboration with the Weiss Lab. And then the second talk will be by Amy Ikui. And this is a project that basically they have worked on really hard, and we were glad to be able to jump on last minute and contribute a little bit, but it's been also a fun collaboration. And so let's start with Marina. So Marina is a PhD student in my lab. She's actually joined the lab during the pandemic, and she is originally from Villach, which is a really big city close to Zurich. She has been very interested in how cells, she joined my lab to kind of understand how cells control their density. And she's worked on multiple projects, but the project she's talking about today is really a collaboration with the lab of Carsten Weiss and there with a PhD student there, Lorena Kronig. And so without further ado, Marina, I will hand over the word and the stage to you.
Thank you, Gabriel. Can you all see my slides? Okay, perfect. So thank you so much for having me to talk about my PhD project here. That's about how pKa regulates biophysical cytoplasmic properties to confer stress resistance through glycogen and trehalose production. And so like Gabriel mentioned this is a collaboration with Lorena Kronig and Carsten Weiss, and we're hoping to publish this soon. So keep your eyes peeled for that bioRxiv link, hopefully soon. So all life had to evolve to deal with stress. And to deal with stress, it has often happened that it was just more convenient to create a unified stress response rather than having many individual ones. So for example, in budding yeast, which is our model organism, this is the environmental stress response, and a lot of different stresses activate this response and then have a stereotypical unified response to it. And a reason for this is that oftentimes not one stress happens at once. So in German, we have a saying, ein Unglück kommt selten allein. One misfortune rarely comes unaccompanied, or in English, you would say, when it rains, it pours. So chances are that while you're dealing with one stress, another stress will come up, and then you don't have the means to deal with this. So it's just more convenient to have one response to deal with all of them. And so even though this is one response to many stresses, the cells respond very differently when you look at their physical cytoplasmic properties. So cells usually, when they're happy and healthy, they have a very narrowly distributed density, crowding, and diffusion regime. But then when they become stressed, two things can happen. Either they become less dense and less crowded and more diffuse. This happens, for example, during amino acid starvation or during hypoosmotic shocks. Or they can become more dense, more crowded, and less diffusive. And this happens, for example, during carbon starvation, around heat shock, or hyperosmotic shocks. I've grayed out the osmotic shocks because we wanted to know how cells regulate their density. And in osmotic shocks, it happens mostly through influx or efflux of water. And so we wanted to focus on the other components. And on the left side, about amino acid starvation, a lot is already known what is causing this. And one main culprit here is TORC1, which is inhibited by this. And this deals with ribosome biogenesis. And this leads to a decrease in cytoplasmic crowding. On the other hand, not as much is known, but we were thinking, where could we start looking for this? So a main thing that happens upon carbon starvation or heat shock is that PKA is inhibited, and that then leads to the activation of the environmental stress response. So we thought it could be interesting to see what happens if we mess with this, especially because there's also crosstalk. So really both things could happen here. PKA is a master regulator of glucose sensing and carbon metabolism, and it also keeps the environmental stress response inactive while there is good glucose conditions. And so we decided to inhibit PKA with an analog sensitive system. So the PKA isoforms are mutated so that this analog binds there in their ATP pocket and makes them not function. And this was emboldened, and this is why we started this collaboration, because the Weiss Lab was working on something similar, and they recently published a very nice paper that implicates the PKA pathway and the environmental stress response in both biophysical property regulating and in survival in quiescence. But now, before I tell you what happens when you inhibit PKA inhibition, I first want to tell you how we are measuring density, diffusion, etc. So one way we measure it is by density gradient centrifugation, which is a very old and familiar concept to many of you. But we are doing it in microcapillaries to make it more quantifiable. And I'm not going to be showing much of this data, but I just wanted to mention it in case any of you would like to have an easy readout in your lab. This is quite easily done without too fancy machinery. But then we are also using to determine density refractive index tomography, where you technically measure the refractive index of any point within a cell, but the refractive index correlates with the dry mass density. And so like this, we can calculate the dry mass density with subcellular resolution. And it also allows us to segment out the vacuole of yeast cells, which is very empty and less dense, and therefore would otherwise skew our data. And then finally, we are measuring crowding as a proxy of diffusion through gem tracking, which is where you track these bright spots inside of a cell, and then you can reverse calculate through the mean square displacement the diffusion coefficient. And it was mostly Lorena that did these measurements. And so what happens if we inhibit PKA? I'm showing you here the refractive index tomography pictures, and you can see here that the dry mass density of PKA inhibited cells increases immensely. And this is all dependent on the environmental stress response, which is downstream of PKA. So once we inhibit the main transcription factors of the environmental stress response, which is MSN2 and 4, this density increase does not happen. And here we have this quantified, where you can also see that the density increase of PKA inhibited cells eventually plateaus at around a 50% dry mass density increase, which is quite substantial. And so it comes as no surprise that the diffusion in these cells decreases dramatically because they are so crowded and so dense. And this is again also dependent on the environmental stress response. We also looked at the confinement of the trajectories, which can tell us something about how the makeup of the cytoplasm. And we saw that there's both more confined trajectories, and they are confined in a smaller space. So the radius of confinement is smaller in PKA inhibited cells, and it's again rescued by MSN2/4 deletion. So we have these cells that are overproducing something, so they have a 50% increase in dry mass density. But what are they producing? So we tested a couple of things where I won't be showing you data, but you'll just have to trust me on this. We find that it depends on the presence of glucose and translation. And we also do not see a protein increase in the concentration of these cells that could account for this massive density increase. So we had to take a step back and see what happens with the other roles of PKA and the environmental stress response. Because among the genes that are expressed upon the environmental stress response are those that lead to an accumulation of glycogen and trehalose, which are usually seen as storage carbohydrates. And indeed, we have a massive accumulation of both of these storage carbohydrates in PKA inhibited cells. So what we can say so far is that upon PKA inhibition, the cell becomes more dense, more crowded, and less diffuse, and there's an accumulation of both of these storage carbohydrates. And this was promising because both of these storage carbohydrates have been implicated in increasing viscosity upon various stresses in budding yeast. This increase and accumulation of these carbohydrates was again dependent on the environmental stress response. And just to be sure that we're actually measuring what we're measuring, because sometimes people say, oh, glycogen can also accumulate outside of the cell in the cortex, for example. We did cryo-ET, and in this we saw consistently in PKA inhibited cells a central clump of what looks a lot like glycogen granules. It's a little bit pixelated here, but you can see this pattern. And we are extra confident that this is glycogen because we do not see this in wild type cells or in cells that are mutated, so they cannot produce glycogen. And in these cryo-ET images, we also saw other interesting things. For example, they have a thicker cell wall, and this is going to be extra interesting when you hear Amy's talk later, so you can keep that in mind. And we also saw other phenomena, like, for example, the vacuoles become smaller and fragmented. Something similar happens with the mitochondria. And the nuclei sometimes are misshapen and almost deformed when there is something next to them. Like you can see here, there's a, I don't know, vesicle of sorts next to them that deforms the nuclei. But so we're fairly certain that what we're seeing is an increase in density because there's this like, as well, we have an increase in density and we know these two carbohydrates accumulate, but does this also explain this density increase and diffusion decrease? So for this, we created glycogen synthesis mutants to see if both of these things still happen. And first of all, if we look at density, the glycogen synthesis mutants have a decreased increase in density, but it doesn't restore them to wild type levels whatsoever. So they only have a mild effect on density. But now if we look at diffusion, something interesting happens because they can completely rescue diffusion back to wild type levels. So what we know so far is that the crowding in these cells happens mainly through the accumulation of glycogen. But now what is up with all that trehalose? Is that maybe the missing puzzle piece? Or what does that do? So first of all, if we look at trehalose synthesis mutant, we cannot do this in fermentable growth medium because these mutants are not viable in those mediums. So we are using glycerol instead of glucose. And their wild type cells already have an increased base level of glycogen and trehalose in them as is, and also the increase that we see is less strong. Yes, then we managed to make these mutants that completely get rid of trehalose, and they're measuring some sort of glucan, but we're pretty sure it's not glycogen again because we showed that there's no glycogen with cryo-ET, so we're guessing this could be something like, for example, cell wall that we're finding. When we look at the dry mass density imaging, it's not immediately obvious because of the large vacuole, whether they're increasing their density or not. So we look at the quantification. And first of all, we see in the pink line, the density increase is much smaller than in glucose. And then what we can also see is that the two glycogen mutants here in orange and blue do not increase density at all in glycerol. And that trehalose does not seem to have an effect on the density whatsoever. So then we look at diffusion, and here it gets really interesting because we actually see an exacerbation of this diffusion decrease in trehalose mutants. And we think that trehalose could therefore have a fluidizing effect. And this is backed up by findings that trehalose can basically form a hydration shell around macromolecules. So we think that once trehalose is not there to fluidize the cytoplasm, we see this further decrease in diffusion. And the double mutant is again mostly fine, similar to only a glycogen deletion mutant. And so this leads us to believe that without the glycogen crowding, you don't need this extra fluidization because, of course, in wild-type cycling cells, you don't have a lot of trehalose and you're still pretty fluid. So you might not need it unless you become very crowded. And that might be the reason why they have to upregulate both at the same time. And so I started this talk talking about stress, and now I want to come back to stress because with these PKA inhibited cells, we have some cells that have an increased density, increased glycogen and trehalose. And this is very reminiscent of yeast spores. And yeast spores, of course, are known to survive strong stresses that would otherwise kill a cell. So for example, they can survive very hot heat, and they can also survive a long time without an energy source. And we would now like to know if the PKA inhibited cells also have this advantage and if it has anything to do with the presence of these carbohydrates or whether it's or the density or not. So first of all, we did a very extreme heat shock. So we went from 30 degrees to 55, which, as you can see from the spotting assay, kills all wild type cells. But PKA cells are perfectly fine. That's why we had to go so high, because otherwise just all the PKA inhibition mutants were just all fine. So this increase in heat resistance is again dependent on the environmental stress response, but it doesn't seem to be dependent on glycogen production. So when we do this all in glycerol to check for trehalose, first of all, we see that already wild type has a much higher resistance to heat stress, similar to PKA inhibited cells. And then on top of that, we see that trehalose mutants do not survive the heat stress. Glycogen mutants are again fine. And then the double mutant is, somewhere in the middle, I guess. But it is known that trehalose mutants have a decreased heat survival. Next, we checked complete energy deprivation, and we do this by starving the cells of carbon. And on top of that, we inhibit respiration with a drug, antimycin A. So this is, again, a spotting assay where we follow the same culture of cells over a long time, and we spot it every week, basically, and see how long they survive in this complete energy depletion. And we can see that the PKA inhibited cells survive much longer, so they survive more than 20 days, while the wild type cells don't even make it to 1 week. This is, again, dependent on the environmental stress response. And if you mutate the glycogen synthesis pathway, this prolonged survival is decreased. When we look at this in glycerol again, the wild type already has an increased survival compared to in glucose, but still PKA inhibited cells survive much longer than that. This is again dependent on the environmental stress response. And both glycogen and trehalose deletions have an impaired survival. And then the double mutant is just as badly off as the environmental stress response mutant, which could mean that really all the benefit they get is by having glycogen and trehalose. So you can think to yourself, okay, of course they'll survive starvation for longer if they have storage carbohydrates, because it's basically like them packing a big picnic that they can eat then over time. So we wanted to know if that is the entire story. And so our first hint that that wasn't the entire story is that PK inhibited cells actually within the first day degrade most of the glycogen they have accumulated. And then secondly, we created glycogen and trehalose degradation mutants so they cannot degrade both of these storage carbohydrates for food or energy. And this stopped the glycogen degradation. And now when I show you the survival spotting, it's very evident that they do not need to degrade either of these storage carbohydrates in order to survive this long. So it's really the presence of these molecules rather than them being able to use them for energy. And this was very exciting to us because, of course, this could mean that they really alter the cytoplasmic properties, and that is why they have this increased stress tolerance. And so this already leads me to my summary. We show that upon PKA inhibition, cells increase their density and crowding, and they decrease their diffusion. and they accumulate large amounts of both glycogen and trehalose. We also showed that glycogen is what makes up most of the crowding of these cells, and without it, they diffuse normally. We also showed that trehalose has a fluidizing role in these cells, and without it, diffusion decreases even further. And we showed that in a double deletion, the diffusion is also normal, meaning that the fluidizing role of trehalose is only necessary in the crowded conditions of accumulated glycogen. Then we show that PKA-inhibited cells have a massively increased resistance against heat stress and long-time energy deprivation, and that this is not dependent on the catabolism of these molecules. This heat resistance persists in glycogen mutants, but the energy depletion resistance is affected. It's also affected in trehalose mutants, and they do not have the temperature resistance anymore. So the temperature resistance really comes from the trehalose. And then finally, in the double deletion mutant, they also cannot survive the energy depletion, and they have a strangely intermediate temperature resistance, but they are also quite impaired in that too. And then finally, I would just like to highlight what a nice tool the PKA inhibition system is, because it allows us to really modify the crowding, density, and diffusion in vivo in a stable manner. So it plateaus after a couple of hours and then it stays quite high. It's also, what I haven't shown you, entirely reversible. So you wash out the inhibitor and they just go back to cycling within an hour. And they also decrease their density, etc. And then we have also shown some conditions in which we can decouple density and diffusion. So for example, the glycogen mutant still has a high density, but it has completely normal diffusion. Or the trehalose mutant, which has the same high density, but even lower diffusion. So if this is something that you're interested in, this is a very nice system that's also very easy to handle. And with that, I already want to thank everyone who worked on this. So specifically, I want to thank Lorena, who was my partner in crime on this entire project, and with whom we're hoping to soon publish a manuscripts together. Of course, also Carsten for all of his fantastic input. Then we had Martina from the Peter Lab do the cryo ET for us, which was very insightful and helpful. And then also, the entire lab and Gabriel and the entire institute. And of course, all of you for tuning in, better late or early, depending on where you are. And I'm happy to take questions.
All right. Thanks a lot, Marina. I was just trying to find the reaction button, but I always fail with this. Yes, what we usually do is we take questions, we type them in the chat, and since we have two speakers, we will only have time for a few questions, but if there are still remaining questions, we can take all the questions also after Amy's talk. And so you can either type a question in the chat. Oh, now I see the chat. Or you can raise your hand, and I think Yuping has a question.
Hey, Marina, this is a beautiful talk. Thank you. I have a question regarding the trehalose. Do you know how it is protective against heat and low energy? And then in terms of the fluidization, I think you cited the paper from Laura Pawson on Brentman.
Yeah.
They sort of show that the trehalose is sort of inducer of phase separation. Do you think that could be an alternative explanation for the fluidization that you see in your cells? Thanks.
Yeah. So thank you. Those are good questions. For the first question, so it hasn't been shown that it is protective against heat stress because it stabilizes proteins, because it is a cosmotrope, so it binds water better than water, and it also binds proteins better than water. So it can kind of donate these hydrogen bonds and therefore stabilize proteins. And this is one of the big issues in heat, is that your proteins get destabilized. Against low energy, it's not necessarily known to be too protective, but it is known, for example, to be required to refluidize the cytoplasm after you exit from being a spore, so that you like refluidize your cytoplasm. That cannot happen without trehalose. And then the other question about the aggregation and condensation. So we haven't looked at specifically what gives it this fluidizing role. We just called it fluidizing, but we do think that there is actually like an aggregation happening because, right, we do that in vitro a lot. We increase crowding and that leads to aggregation. That's essentially what we're doing here. I can absolutely see that happens. And the interesting thing, the difference between our findings and the paper from Laura Persson was that they kind of threw trehalose and glycogen into the same pot. And we now actually see that they have very different roles when they accumulate.
Thanks.
Then we have a question from David.
Hi, Marina. Really good talk. Cool
Thanks.
talk. I wanted to ask you if you mentioned changes in the diffusivity, if you maybe see some changes also in the alpha value, when you fit the diffusion equation to the anomalous diffusion and you see changes in the alpha value, like trying to see if there's not only changes in the way the particles move, in how fast the particle moves, but also the way they move. Have you explored that?
Yes. So very sadly, Lorena, our expert on this, is I think not here today, but I'll try to answer as good as I can. So we looked mostly at the confinement, which of course inside of the confinement, they can still diffuse as fast or as unfast as they can. And so we usually used confinement kind of as a proxy of how the cytoplasm is organized. We never looked specifically at subdiffusive or like super diffusive behavior, mostly because it's also kind of like a definition question, right, with like the resolution you have. But it is interesting. So both in the confined and in the non-confined tracks, we see a reduce in diffusion. So it really is not just that there's more confined tracks and therefore the whole diffusion is lower, but it's really every part of the cytoplasm is affected by this, if that makes sense.
Yes, thank you.
Thanks.
Thank you, David. I think maybe I can add to this from Lorena's. I think so. They see this increase in confined tracks, and I think that's in part defined by an alpha value threshold, which is kind of reflecting. I think that there is a kind of that the alpha is affected in a way. Then we have a question from Hyojun.
Yes, nice talk, beautiful talk, Marina. Thank you so much. Would it be possible if the nuclear plasmic diffusivity could be controlled with similar manner with PKA? I know the site is mostly is known to be located in the cytoplasm, but as a smaller molecule, it could be any possibility they get aggregated in the nuclear plasmic area. So would you have any clue about it?
So that's a really good question. We were thinking of opening that can of worms, but no longer for this project. So I definitely think it'll affect the nucleoplasmic diffusivity, partially because, of course, like you say, trehalose is a small molecule that can diffuse pretty freely and will affect the nucleoplasm. And then also we see these compressed nuclei, right? So chances are that they are also denser. We don't see them to be much less dense in the dry mass density images that we do, right? So I'm guessing that diffusivity there would also be affected massively. So if that is something that you're interested in studying, I'm sure the PKA inhibition system would also, be very interesting.
Thank you. Just last quick question about how can I say? Have you checked the EM image in the nucleoplasmic area? I think it's visible in the nucleoplasmic area.
We haven't checked it so much. We also don't necessarily see a huge increase in electron density overall, but we are doing in situ cryo-ET and not TEM, where these changes are just not as evident, but it does allow us to see like the whole cell. So we decided for that because we're being very exploratory. But I'm sure you could see this easily.
Thank you so much.
Thank you.
So I think in the interest of time, thanks, Hojun. He's very interested in the nuclear things because I was just recently on his defense, which was really beautiful, by the way. So I think in the interest of time, we will save Tomoko's and Jinrong's question for later, if that's okay. And then we would move on and give Amy the stage. So maybe a quick introduction. So if I got it right, Amy was born and raised near Kobe, I learned recently, in Japan.
Born in California.
Oh, born, sorry, see, now. Okay, well, but there's the connection to Kobe because
Yes.
she,
I grew
figured,
up in Kobe.
okay,
Yes.
Kobe. And that's where the beef is from.
Yes.
Okay, the Kobe beef, the
Yes.
famous. And then for your PhD, she moved to New York City at the Albert Einstein College. And then for the postdoc, she was with Fred Cross at the Rockefeller, where she got really into quantitative biology. And her lab has been really interested in that she opened in 2008 at Brooklyn College. She's been really interested in cell cycle control, both in yeast, but also in Chlamydomonas, which is a really interesting model organism. But then her lab also became really interested in size control and density control. And that's something we've been talking about for a couple of years already and on multiple conferences. And so I'm really happy to see how this project has evolved over the years, and that we were also able to be play a little part in it. And so without further ado, thanks for coming here and for presenting this beautiful work.
Thank you. Okay. So now I'm talking about different kinase. So after Marina's talk, and I will talk about the MAP kinase during the cell growth. Oh, super slow. Does it change my slide or no? Oh, here. So our lab isn't interested in the large cell. So as you know, within the given cell types, cells maintain the normal cell size. But what happens if they grow beyond the normal cell size? So we can force yeast cells to become extremely large using the cell cycle mutants. So for example, G1 arrest is very efficient to make super large cells after long G1 arrest. And in those large yeast cells, we know that total RNA protein concentration go down. It's a cytoplasmic dilution. And then this type of dilution is a hallmark of aging and senescence. And when cell size is larger, the cell proliferation rate goes down. And we also know that environmental stress response proteome goes up. And we already learned about it from Marina's talk, such as pKa. So obviously, cells know their cell size. And I have been asking this question, how do cells sense the cell size when they become large? So that was the big question in my mind. And when I think about the cell membrane and cell wall, maybe cells have a system to sense the membrane stress, and they may activate the signaling pathway. So first, we know from a fissionist and mammalian system that when cell size is larger, the cytoplasmic dilution happens, and we can use GEM to measure the diffusion coefficient. So I also used GEMs. My GEM is 40 nanometer GEM. I assume it's same as Marina's GEM, and the size is equivalent to ribosome. So the very first experiment, we used small cell mutant V5, large cell mutant CLIN3, and then measured the GEM diffusion coefficient. And as you can see here, this is after 3 experiments. The large cells in CLIN3 mutant, GEM diffusion increases. And this is a single plot. So V5 is plotted as orange, wild type gray, and CLIN3 in blue. And we plot cell area in the x-axis versus y-axis is GEM diffusion. So you see this graph through my talk to show the GEM diffusion versus cell size. And then next, so we also used other cell cycle mutants to confirm this. And in order to regulate the cell size over the time, we used the conditional mutant of CD28, CDK1 homologinist. And this is analog sensitive mutant. So if we treat cells with the 1 NMPP1, cell size become bigger, as you can see here. We also treat cells with the cycloheximide to reduce the cell size, which is our control. And then we also confirm that GEM mobility and diffusion rate increases with the 1 NMPP1. And this is a single cell plots over the time from green time 0 to 8 hours. So, and everything goes back to normal after cycloheximide treatment. And this is the movie. Even without the analysis, you can see that it's much faster after even 4 hours, the GEM mobility. So going back to my first question, what kind of signal pathway is activated? So my lab has been studying this cell wall integrity pathway, and my previous project focused on how this pathway inhibits the cell cycle. And historically, this pathway has been well studied. And we know that upon the cell wall damage, usually people use SDS treatment or laser to damage the cell wall, activates RO1, protein kinase C, PKC1, and then downstream kinase is MAP kinase. So this MAP kinase MPK1 is the very downstream kinase which directly activates the transcription factors. And then basically activate the transcription of cell wall biogenesis, and then make more cell wall components, repair the cell wounding after the damage. So this is classic signaling pathway that we know of. So my question is, when cell size increases, maybe something happens to this cell wall or plasma membrane may activate this signaling pathway. And I was a little bit struggling with this. My idea already has been there maybe like 8 years ago. And I have been talking to people. And I remember probably during the COVID, I was talking to Gabrielle. We did a lot of Zoom. We had some extra time during the COVID. And then he said, oh, I think we got MPK1 in our phosphoproteomic analysis. So this is his data. Marina, Federico, and Ruth were involved in this experiment. So in this experiment, they used CD28-13 temperature-sensitive mutants, obtained the sample 2 hours, 4 hours, 6 hours after the temperature shift. And they made 8 different clusters based on the patterns of the changes in phosphoproteome. And if you focus on this cluster 4, this cluster gradually increased in the phosphorylation levels. And when we closely look at this cluster 4, the top hit was MPK1 MAP kinase, which means that MAP kinase substrates tend to be more phosphorylated upon the increase of the cell size. So we asked Federico to look at more in detail. So we observed that MPK1 abundance also increases over the time. So this is a protein level. And then also MPK1 is autophosphorylated when kinase is activated at T119, Y192 sites. And this phosphorylation level is also slightly increased, which implies that probably MAP kinase, both protein level and kinase activity is upregulated over the time. So we decided to follow up on this. So our postdoc Keiichiro has been working on this. So this is the summary of cell wall integrity pathway. MPK1 is a very downstream. And we know that MPK1 is translocated to the nucleus, activate the transcription. Main target is RLM1, which make more cell wall components. So we made an MPK1 neon green strain, and this is the 8-hour time course. So in the wild type at time 0, you can see MPK1 both in the nucleus and cytoplasm. The total level increased over the time, and also MPK1 protein intensity in the nucleus also increased. And this is a NAP49 nuclear marker, and this is the quantification from the images. So this is total MPK1, nuclear MPK1 intensity, and this is a nuclear to total MPK1 ratio, also increased over the time. We also deleted upstream kinase BCK1, and then observed that MPK1 intensity decreased as well as nuclear intensity, and this is nuclear to total MPK1 ratio. So probably this whole cell wall integrity pathway is activated upon the cell size increase. Then Keiichiro is a very careful person, so he was a little bit worried that, oh, maybe all other proteins may be increased. So we looked at the histone, HTB1, which is known to be diluted over the time by proteomic study, but I think nobody actually looked at it under the microscope. So this experiments, it nicely capsulated that nuclear size also increased when cell size increased. And then in the CD20AS1 mutants, MPK1 is accumulated in the nucleus, but this histone proteins, it's diluted. So total level remains the same, but because of the increase of the nuclear size, the protein is diluted, and this is a quantification. So definitely MPK1 level is upregulated, and also in the nucleus. So important question is what happens if we delete MPK1? So interestingly, if we delete MPK1 from this AS1 mutant, cell size become even larger. It's like a huge balloon. So this is a cell size. The double mutants is in blue, and the orange is S1 mutants. Diffusion coefficients become higher in the double mutants because of the cell size increase. And this is a single cell plots. It's quite remarkable that this double mutants, the cell size is much larger, and then diffusion also faster. And everything go back to normal with the cyclohexamide treatment in the double mutants. So how about other cell wall integrity pathways? So we used other deletion mutant from the deletion libraries. So if we combine BCK1 deletion with the AS1 mutants, cell size become larger, gem mobility becomes faster, as well as for MIT2 and WSC1, which is the cell wall sensors. we didn't observe difference in other cell wall sensor, probably because of the redundancy, but this RM1, but very downstream transcription factor itself, if we delete this factor, both cell size and gem diffusion becomes larger. So it seems like whole pathway is involved. So what happened to the cell wall, right? Marina also showed that some cell wall thickness in the PK1 mutant. So in this large cell mutants, the cell wall also much thicker. So this is the plasma membrane here. Cell wall is thicker than the wild type. And if we further delete MPK1, because MPK1 function is to make more cell wall components, so cell wall is very thin, and this is the cell wall thickness measurements. So you can also see other very interesting other features, something going on, a lot of lipid accumulations. Vacuole is huge, and this W mutant, it seems like something is going on in the vacuole. So, but I'm not going to talk about it. That's something we want to continue. So then Gabriel brought us this Laplace law that probably because we measure the cell wall thickness, we can apply our data to this equation, and Leo took care of this project. So the equation is this tensile stress on the cell membrane can be explained by internal pressure times cell radius, so basically cell size, divided by cell wall thickness. So when we apply our data to this equation, so this is the raw data, cell wall thickness increases as cell size increases. And then when cells do not have MPK1, they have trouble making cell wall. And then if we apply this data to this equation, the relative pressure for these AS1 mutants, they maintain the same pressure because of the thicker cell wall. But in the absence of MPK1, they fail to maintain the pressure. So this is nice Gabriel's cartoon. During the active growth, AS1 mutants, they make thicker cell wall. So cell wall is reinforced to maintain the pressure and crowding. And if there's no MPK1 or other cell wall integrity pathway, they probably make equal amount of components. However, cell wall is stretched and thinner, which decrease the pressure and crowding. And then this Laplace, this is the Laplace, 1804. So this is kind of a beauty of the science that we can prove his equation, mathematician's equation, after how many years? 200 years. So, so I'm running out my time, so I quickly finish. So what happens, right? Cell wall is thinner in the absence of MPK1 in large cells. So probably we can prove this using zymolase to digest the cell wall. So Keiichiro observed burst cells or bleeding, which means that they try to form the bud but fix the problem, spheroplast. So this is high concentration of zymolase, and then we treated these cells with a very low concentration of the zymolase. And if you focus on G, this large cell itself somehow a little bit sensitive to zymolase. So this is the percentage of non-burst cells. And then this double mutant super sensitive to zymolase, which makes sense. And then this is accounting. Most cells experience burst or bleeding morphology after the low concentration of zymolase. And without the zymolase, if we try to exit from cell cycle arrest, so we removed this 1 pp1 after 8 hours, wash out, let them go. So this cell cycle arrest itself is reversible. So this is a serial dilution data that they can grow back to normal if we wash out the cyclohexamide. But if we remove MPK1, they have a hard time to regrow. And then if we take the images, they just burst. They can't tolerate with the cell cycle restart, probably because of the cell wall thin problem. Okay, so with that, the summary, we use cell cycle arrest to make cell size bigger. They reinforce the cell wall due to the cell wall integrity pathway. And then these are the main components involved in this large cell response. And then actually we just submitted our preprint to bioRxiv. So if you are interested in, you can take a look. And then this work is mainly done by our postdoc Keiichiro Sakai, who recently moved back to Japan, and also our lab members, and of course our collaborator, Gabriel's lab member, Marina, Federico, Leo, and Ruth. And TM was done in Japan, led by Yoshiaki Kamada. And then also GEMS coming from Liam's lab at the NYU and funded. And I'm happy to take questions.
Thank you, Amy, for a
Thank
wonderful
you.
talk. No, I found it. So yes, so the same procedure as before. If you have questions for Amy, please put them in the chat or raise your hand. And maybe.
So maybe I can make a question.
You're right. No. See, Hujon. Also, sorry, I'm still butchering your name. I'm very sorry. I know who you are.
I know who
Already
you are
has a
too as
question.
well, so it's a
Okay, good.
mutual, so
Good.
perfect. Thanks, Amy. Thanks for a nice talk. And I was so grateful to host the video, the preprint the last week. I had some like basic questions about the data. I was wondering I don't exactly remember the pages of the slides, but in the figure 4, I found that once the 1 pp1 like induced the volumetric growth of the cell, the gem diffusivity, I would say the diffusivity increase got accelerated pretty faster than the volume increase. I was thinking it could be not only due to the Zürich equation and the only physical changes, but also it could be due to the polysome collection or like glycogen reduction, the other glycogen changes. So
Yes.
do you have any other explanation about this kind of let's say?
Right. So definitely,
yeah.
right? If they have problem making messenger RNA, or then maybe polysome level decreases with ribosome. Not if ribosome biogenesis has some defects, so that could be the case. We didn't measure glycogen, but we measured the trehalose, and there was not much. Keiichiro was trying. So work was to study trehalose in spores. So he looked at carefully using trehalose mutant, but we couldn't see much difference.
Okay. Thank you.
I don't know about the polysome. Yeah, definitely that could be a very possible point.
Yeah.
And I remember you asked the question about the nucleoplasm gem for Marina, right? So we looked at nuc gem. Nuc gem is in the cytoplasm in these mutants.
Okay, thanks a lot.
We don't know the reason why.
That's so kind of you.
Yeah. I thought maybe proteins are leaking, but it seems like proteins not leaking, so maybe they have some assembly problem in the cytoplasm. So definitely that could be related to probably polysome levels. They have issues making proper proteins, could be, even dimers. So yeah, everything is kind of messed up.
Just a very, very small suggestion that I had about the paper. Like you used the cyclohexamide to, let's say, stop the whole translation event. Maybe how about to use the lactidomycin to like relax the whole, like to let the polysome, let's say, to prevent the polysome freezing up. How can it? I don't know. Yeah, to prevent the polysome collection.
Yes.
So that's one small suggestion I had. Thank you so much for your talk.
Thank you.
Great. And then next up, we have a question from David.
Hey, Amy, really good, cool talk. Can you say hi to Keiichiro?
Yes.
I was wondering, you said that enlargement is a hallmark of senescence. And somehow when I see cells aging and dying, that's what I see. They get really, really, really large and then they kind of burst. That is kind of what you're showing here. So do you think there's some connection between these and the cells aging and dying?
Yes, definitely. So I really want to know that answer. So if if you measure the cell wall thickness, maybe cells are trying to reinforce everything in the aging cells, and some point, maybe later lifespan, they can't maintain that cell wall physiology, and then they may burst. So I'm trying to believe that this is same as aging cells. And then, I looked at the recent Micron's preprint comparing the mass spec between the aging cells and large cells. Many proteins are overlapped, including MPK1. So yeah, that's something I want to pursue in the future. And collaboration is welcome.
Okay.
I don't know how to collect our aging cells.
Yeah, sure. We can talk. Yeah.
Thank you.
Good. And then Jinrong, whose question unfortunately before wasn't answered, but Marina was so kind to answer it by writing. He has a question about NOD49.
Yeah, thank you. Just about the when the cell enlarge, you see an increase of new 49, both the total amount and mean intensity. So and it's different from the H2B intensity, which is diluted. So I don't know why. Do you think it's because of the when the cell enlarge that you have actually more nuclear pore complex assembly there?
So that I'm also very interested in. So I'm planning to do super resolution microscope for this. Either numbers are changing or just each pore, they have more proteins. I think something must be happening at the nuclear pore, right? Because it's same as a plasma membrane. Nuclear membrane also stretched out. So that's also another good research area. And by the way, I'm looking for postdoc.
All right. Yeah, thank you.
Yes, thank you.
Thanks. And then Tomoki, I'm confused. No, Tomoki also has a question for you.
Okay. Vacuole size, yes, definitely. I think vacuole is some type of compensation mechanism. So her question is, vacuole size is increasing in large cells and it's related to maintain the cytoplasmic concentration. So probably, so vacuole is a smart compartment that probably less understood in this area. So definitely, I think they are trying to reduce the dilution by making large vacuole, and the vacuole is gigantic. So yeah, definitely. So we can easily use vacuole mutants and then make vacuole smaller, and what happens? So that's on my grant application.
I see. Oh, that's really cool. Yeah. It's interesting to me that the components of the cytoplasm and vacuoles are very different, but yet probably the whole cell and vacuole can regulate their sizes. But yeah, maybe potentially independently or maybe with some crosstalk. So I think that's very very fascinating. Yeah,
right.
thank you
Might be iron, right? The vacuole uptake of the water from the cytoplasm, and then but we don't know the mechanism how they sense that.
I see. Thank you so much.
Thank you.
Good.
Thank
So
you. A lot of
yes,
potential projects.
exactly.
Gabriel.
So if there are no more questions, then we will finish for today. Thanks all for joining and for sticking around for the questions. And I hope to see you back here on either August 19th or September 2nd, and you will get an email from Yuping about this. And so thanks and have a good summer.